to present basically some options that we have to evaluate clustering with weighted nearest network. That is a cross-modality to, in this case, it's applied to integrate data for single-cell ATAC-seq and single-cell RNA-seq. So briefly, I'm going to explain a bit of background. I'm expecting that we could cover, let's say, at least an overview of what is the weighted nearest network for multimodal analysis, as well as a glance of what is the clustering algorithms that we have available in scRNA, and some clustering evaluation methods that I found useful. These are not the only one. There are several methods available over there, but these are some of the let's say the strategy that I follow to explore my data. So just to start, I would like to remember that in this kind of, we have a multiome of single-cell RNA-seq data with single-cell ATAC-seq data. When we are expecting to analyze together both modalities, so we want to connect the gene expression with the accessibility chromatin information in order to have clusters that represent both modalities in just coming from the very same cell. So that way we can have the opportunity to look for cis-regulatory elements or try to describe regulatory networks. So in the weighted nearest network clustering, using this kind of data with scRNA, the big picture is that first we pre-process the data. It means that we count the data and then we apply normalization, we scale the data, we apply then the RNA data and the ATAC data are like prepared separately. So for RNA data, we apply dimensionality reduction and we can make some data correction. And for ATAC, it's applied the LSI approach that it includes 2 or 3 steps that I'm going to talk about in a minute. So the last step when we are making clustering with this kind of approach is to perform multimodal clustering when we want to integrate the data from these 2 modalities. So we need the dimensionality reduction assays in order to do that. 2 things that is useful to understand when we are working with this is that for ATAC, the most broadly used approach is the Latin semantic index. This is a linear dimensionality reduction method that was originally used for text mining. But in the context of ATAC, it transforms the sparse and high-dimensional ATAC data into lower-dimensional space to capture important parents. So it's a way to say that it's analogous to the PCA for RNA. So how it works, basically, it has 2 steps. One is the term frequency IDF transformation that basically is normalized using this approach to adjust for biases. And the second step is apply the singular value decomposition that is applied to the TF-IDF metrics to identify components that capture the most variance in the data. So at the end, the output is this reduced dimension that summarize the chromatin accessibility patterns. So all this is working separately for ATAC. Now, what happened with the RNA? We apply the PCA, let's say a regular PCA, and we can make any data correction if necessary. So when we want to call the modalities, using the weighted nearest network clustering, at least in scRNA, all the algorithms that scRNA have available uses a graph-based clustering approach. So let's say that the main goal is that we have as input the dimensionality reduction for RNA and ATAC, and we want to have the cross between these 2. So in the middle, in scRNA, we have available 4 algorithmic methods. One is original Louvain. Then we have Louvain multilevel, that is a kind of approach that is trying to correct some of the fails that original Louvain have when we have very big data. Then there is another one, is called small local movement. That is, it's not widely, let's say, benchmark. So that's the reason why it's not a lot used, but I think that it works well. And the fourth one is Leiden. The Leiden, well, is, let's say, it's another approach that is trying to assure that all the nodes in the community are connected. So the methods indeed embed cells in a graph structure. This is the first step. So this is an example. We have an Nails-Nero graph with edges and draw between the cells with similar feature expression partner. And there is an attempt to partition the graph into single interconnected quasi-clickers. That is a way to say that we are trying to have fully connected communities. There are some algorithms that have the ability to have a maximum quasi-click. That is just a way to say that they try to expand to the maximum click that can be identified in this kind of density nodes and edges available. So even when all these algorithms follow the same approach, there are differences in the methodology, in the performance, and in the quality of the clusters that they produce. So this is something that I'm going to talk a bit about it. So I'm not planning to talk about all the details, but these are some of the features that I find really important or relevant when we are working with this approach. So we have here 4 properties. One is the connectivity. It's talking about how the nodes are connected by edges inside a clicker. And this is the quality of the clusters. That is a way to say that if these are fully connected or not. We have also a very relevant feature that is the computer resources that we need. We have a cluster available or not, and the flexibility that has the algorithm or the method to produce different configurations. So here I'm only comparing 2 algorithms. That is the Louvain original and the Leiden that, in my opinion, is like a second generation of algorithms that arises because when grows the number of the databases or the data sets that we have to analyze, it was necessary to have available a different approach that could handle this big number of data. So comparing this, one of the main, let's say, arguments that you can find wherever is that in the connectivity using Louvain, it has been proved or tested that we can have communities that have some of the clusters not fully connected. So Leiden is trying to ensure this connectivity, like adding iterative steps. So it's a way to say that with Louvain, sometimes if the data are not, let's say they have remarkable difference, it can get stuck. So Leiden is trying to extend this concept in order to make partitions according with a different approach to make the clustering. In terms of the computer speed that we need, well, Louvain is very well to make a fast exploratory analysis because it's really fast in the way, and Leiden requires a lot of resources to produce this partition properly. In terms of flexibility, both approaches have enough, let's say, features to try to avoid the over-partition of the data. So we can add, in simple terms, it's like to say that we can increase or decrease the resolution, and we can also like define how many neighbors we want to consider for produce the clusters. One way to summarize this is the Leiden ensure well-connected clusters if computational resources are not a concern. that doesn't mean the Lobain is not working well, because it's really good for a quick clustering on very large data sets where speed is not critical and you are not worried about it. And this is just a kind of metric that I found that is recommended, that when we have more than 100,000 cells, probably you should translate them in order to not have a lot of cluster or communities with nodes not really well connected. So what was my motivation when I was doing this is that when I do the first weighted nearest network using the PCA in harmony with the LSI for RNA+ATAC, I use the Lobain original resolution 1 and the KNN equal to N. This is, let's say, some kind of the default values to make the clustering. So we have here the RNA clusters with Lobain, we have here the clusters with ATAC, and we have here the integration with both of these two data sets. So I noticed that we have a lot of singletons. In Multiome, a singleton is a cluster that we have over here somewhere that is representing only one of these modalities. So it's not a good diagnosis to have a lot of singletons produced, because we don't have the information of both data sets in some of these clusters. So I was a bit worried because of that, because we are losing, let's say, the opportunity to look for regulatory elements when we have this big number of singletons. So I start to check what kind of algorithms we have available, and also what are the main or the more sensitive arguments that affect the clustering. And well, this is well known, the resolution, that is the way to talk about if we want larger or smaller clusters. And also this other argument, there is the number of nodes that we are considering. This is also a very sensible parameter that is affecting the clustering. So here, what we do was to just prepare a script that can run all these combinations. So this in total are 48 different clustering approaches, just to look what are the main changes, and if we have some concordance or what are the discrepancies between the clustering, and if we can cache the biological information that we want to see. So after to have this, there are some clustering evaluation steps that were defined. The first one was very easy, because let's say that the main goal was to identify the weighted network clustering that integrates both RNA and ATAC data while minimizing an excessive number of singletons, because with the default values we have 9. So we are trying to reduce, and we find that we can reduce this to have only 4 singletons. So it was only from the 48 outputs that we have. Indeed, A options, but we reduced it to 4 because let's say that it was a more suitable number of parameters that we can configure to get this clustering. So after to select this in the first step, I really was exploring 2 main resources. That is the orchestrating single cell analysis with the bioconductor book, the version 2025. And okay, there a lot of description that I'm not going to really talk about, but let's say that the main difference that I found when I am applying this to scRNA is that in the book, they are applying all the methods to single cell experiments. So I have to translate my object or just pull the data that I need to reproduce the same kind of evaluation. This is the main step. And I apply only 3 of the available strategies to evaluate clusters. One is based in concordance, based in the Jaccard index for each pair of the clustering outputs that we have selected previously. We do this to look for the best match and observe cases of overclustering among the different methods. The second strategy was to use the silhouette for a proximity analysis to assess the clustering separation and to see if we have community that has a lot of heterogeneity. And the last one, it was to keep a look to the biological signal based on gene expression or canonical genes to try to see if we can catch something and if this is suitable to continue the downstream analysis. So here we have just, let's say, an illustration just to show you that, for example, here, oh, sorry. Here, what we have is low vein, unladen, and we have different kind of resolutions. We have here 20 neighbors. Here we have 40 neighbors, and we have here, again, 40 neighbors, but with laden. So you can see that the number of neighbors affects a lot the kind of way that the algorithm can plot information between the ATAC and the RNA-seq data for this particular case. And also, when we were like analyzing this, we can see that definitely the algorithm, let's say the parameter that most affect the way that we can reduce the number of singletons was definitely the number of the nearest networks to consider. So we find that using nearest network, all the clustering methods, it doesn't matter which of them, if it's low vein, if it's low vein multi-level, laden, or SML, all can keep only 4 singletons identified in the data set. So definitely this tells something about this kind of parameter. At least for all these algorithms that are based in graphs, it looks that it's very sensitive to this kind of resolution. So to summarize this, well, we have a table when we extract all the information for the 48 different runs with the different methods, with the different kind of resolutions, with the different kind of nearest networks. And so we identify which of these have the less singletons, and we extract the information. So is where we realize, okay, the KNN is the critical step for this kind of strategy for this data. So next, the next step just was to annotate the clusters. This is just an example. When we annotate, for example, here, the weighted network with domain. So it just was to try to identify where are the clusters that we are looking at. So in this case, we are looking all the clusters that are identified here with an HB, all the clusters that have an HB referring to Habenula. So most of the clusters are in this cloud, like some kind of mixed, but we can identify small subcommunities in this. So that is why we use the silhouette approximation to try to identify these differences. But this is just to have annotated the clusters in order to make a more accurate interpretation. So this is for laden. And you can see that in all the algorithms, we have the same cloud in the middle, and we have here like 3 small communities in this size. So most of the clusters have the pretty same information represented in the outputs. Now, this is the first evaluation step that we use. That is to compute the Yakkar index for each pair of clusters. So here in the x-axis, we have laden, and in the y-axis we have low vein. So here, this method, what makes is to normalize the differences in the cluster abundance. So that large cluster is not going to dominate the color. So it's a way to make a fair comparison. So one thing that we have to consider here is that route with the strategy that I'm using outputs or arrange the cluster in decreasing order. So the bigger clusters are at the beginning, 0, 1, 0, 2, 0, 3, and the smaller cluster is at the end. So we can see here that the annotated clusters are the first one, and in both kind of outputs. So one of the things that we want to pay attention is that we keep a diagonal that can make, let's say, a projection of the concordance between the cells in the different clusters. And also to see, for example, if for this cluster, that is the cluster 02, and in Leiden, we can see that is split into clusters in Louvain. For example, here we can see that this is split in 01 and 02. So this can tell us a couple things. One, that we can have a kind of overclustering with one of the clusters if there is not a pretty well cell separation, or if these are not subdomains, so it is talking about an overclustering. So there are some things that we were to annotate and to pay attention what was happening between these two approaches. We have the same here, for example, for the cluster 4. The cluster 4 is also split into clusters in Louvain, and it happens the same with the cluster 13. It's split into clusters, the 15 again. And so this is the difference between the total cluster that we have, because we have here in Leiden, we have 33 and we have 37 in Louvain. So at least in this step, we know that yes, there is concordance, there is not a big difference, but there is also a clue that could be an overclustering handled by Louvain, but we don't know in this step. So we do the same with all the different approaches that we select. So we have 6 of these combinations of clusters. We cannot read all of these, but we can see that most of the diagonal keeps in the middle. And we can see also what kind of clustering makes the bigger differences. For example, Leiden against Louvain multi-level, and also smart local movement against Leiden. So, and this is the one that I presented a few minutes ago. So after this step, we want to analyze, let's say, if these cells that are in the cloud that are buried together, let's say, we want to know if it could be some kind of pseudomains or other kind of cells, or the data are so mixed. So in this point, we work with the distribution of the approximate silhouette width across cells in each cluster, and we compute the average distance to all cells in the same cluster, but also in another cluster, taking the minimum of the average across all clusters. So what we have here is that in this plot, this silhouette plot, in the x-axis, we have the clusters, and in the y-axis, we have the measured width. So we have here 0. This 0 is representing, let's say, the limit between the clusters. The cluster that we have with a long tail above 0 are representing, let's say, very specific cell types that are not so mixed with other cell types. The cluster that we have below the 0 are these kind of clusters that the proximity between them says that it could represent, this could be the same, but with subdomains, or it can be mixed. So we have the 2 pictures here. So, but as we have annotated the cluster, we know that most of them correspond to Habenula. So that is why we annotate first, in order to see if these could represent communities. And I don't have here a mix of, let's say, glia with another kind of cells, thalamus or vascular vessels or whatever. So most of them, as you can see, are representing Habenula cluster. So for my perspective at this point, this could be subdomains. So under this kind of evaluation, the most interesting clusters are those that are below 0. So also, like, this is very difficult to read because here, it's not seen here, but there is a scale of color that says something like that. For example, the pink are the most specific cell types. And then the scale starts to change in color. So it's really difficult to try to interpret the colors when we have this kind of projection. So another alternative that you have to do this is just to plot this in a table. So you can plot the silhouette, a silhouette table, when each row corresponds to each one of the clusters from the 1 to the, from the 1, sorry, from the 1 to the 26 in this example, and from the 1 to the 33. So this is the diagonal that is crossing from side to side. So large of diagonal counts indicates that the cells are easily confused with those from another kind of cluster. So we can work and we can know exactly how many cells are inside that, let's say, situation to make a more deep analysis. So we do the same with all the clusters that we pre-select. We work this with Louvain, with Leiden, with Louvain multi-level, and with the SML, and we can see that really it's very consistent analysis, even when we have different clusters, because some of these algorithms tends to produce or to split some of the clusters. Most of the clusters are situated in the first 10, 15 clusters. And also they are like below of 0 in the width of silhouette, so representing probably some kind of subdomains. Lastly, the third step was to see if we have indeed, we can see the expression of canonical clusters for this kind of a study. So we have the PO4F1 and the GPR1151. So here is the number of the cluster, Louvain multi-level, Leiden, and SML. So the green is representing the GPR151, and the red is representing the PO. So you can see that most of the clusters have exactly where it's annotated in the UMAP, the clusters, in the middle here, and we have a few more here in 3 of these small clouds of the information. So it's also very consistent, but it's not enough to see if there is the expression there. So we also use these like dot plots in order to measure the percentage of these specific genes, these canonical genes in the cluster that we have. So we can like have some kind of proof that we have a signal of these particular genes in the cluster that we have. And we can also check that there are particular clusters where we don't have a high like signal of this. So in cases like this, for example, when we have split it here in small clusters, because as we can see, Louvain tends to split the clusters in two. So we have a very small population here that represents less than 1%, that this is slightly seen in the Louvain multi-level cluster. So I think that it was split from one of these clusters here. Also, we can keep a look just to the based in the gene expression. So we have also here represented all the clusters here. This is an example for Louvain multi-level, and we can see the signal of the different clusters from a different perspective. And well, these are like the different ways to identify the clusters. So finally, I'm going to present some of the code to do that in 10 minutes or less. But I want to summarize my notes saying that for the multiome analysis, might be relevant to measure the number of cells that we have per cluster, because we are expecting to have cluster with at least 1,000 cells to identify 80% of the putative cis-regulatory elements. So we need to take this into account when we are working with multiome data. Of course, there are another diagnosis methods to evaluate the clustering, separation, and stability, or methods to compare the clustering that represent different views of data. But these are some of the statistics that I find useful and I want to share with you. And well, I'm going to briefly explain here the 2 or 3 of the relevant steps, not all the code, but just to say that the tool, the package that was used to do this, well, the object is a separate object that integrates the chromatin accessibility information with the combo with snATAC. But indeed, the Weier's to compute the Weier's network is made with surrogate functions, and we use Bluster, particularly to make the Jacquard index and to compute the silhouette approximation. And all the packages is just to manipulate the data. So I'm going to just jump here. This is just to load the objects. I have here a function that is important because this is to prepare the data to plot. Because if you follow the OSCA book, they are working with the single cell experiments. So we have two options here, no? One is to translate the separate object into a single cell experiment or just to pull the data in matrices in order to save memory because I was struggling a bit with the memory resources. So when I was working in this interactive mode, so usually broke because the memory resources. So one way to work with this, because I have to load two big objects, was to just import read the separate object here, and also just to pull the cells that are coming for the integrated harmony. So I have here the metrics, and then I just pull the separate in a simple data frame. So having these two objects, I can compute the Jacker and I can compute the silhouette approximation and whatever method that we want to apply for evaluation. So I think that this was critical to make work well this script. And so I can here load different separate objects and just keep the data frames and not all the full object with all the asides and all the dimensionality reductions it contains. And well, the second step, in all one of the recommendations that I can do is that it's difficult sometimes to read these plots because they are composed of a lot of colors that I really cannot distinguish. So I used to just to pull the data frame and just I save it like as a table in order to go back and forth and check if I'm not just trying to figure out things, but I can see the numbers and see what is the percentage of the approximation of the clusters without guess too much. So here so you basically prepare the information calling the function, and here I call the second clustering to prepare the information. I only have data frames here available, and I call the approximate silhouette function to prepare the data. So it's very simple indeed. And to prepare the Jacker is also use a Bluster. And to use a Bluster, also I'm working only with data frames instead of the big objects, and preparing the columns and the row labels. And finally, here is just prepared a heat map with the data that we have previously here. So something that you can find useful after to prepare all this data, give some colors and all this stuff, is the cluster calls and the cluster rows that as a hierarchical like plot, as a hierarchical tree inside of the heat map. And the other parameter that is useful is to display numbers because you can see exactly the percentage of the concordance between the cells. So it's very useful if you have enough space to see this in a heat map. In my case, it's not possible. There are really small numbers. And yes, that's it. This is all what we need to work with these two evaluation methods here. Yes, that's all what I have. So if there are questions or any comment, yes. Thank you.